So heredity or causes a phenotypic variation, heredity, so the animal's genetic background for phenotype. So we know that the cow and the bull are going to contribute. And then the environment, so conditions in which those animals are raised and born. So it could be climate, nutrition, disease, general management. All of those factors can influence the phenotype of the animal. Heritability is something we've got to think about, and that is the proportion of the total phenotypic variation that is attributable to the additive genetic content. So the degree of the relationship between the phenotype and the genotype. And it's generally reported as a percentage there. So essentially heritability is going to tell us, through genetic selection, how can we make change in these animals over time. Production traits vary in heritability, and most reproductive traits, conception rate, etc., are low in heritability, so less than 20%. Production traits like weight are medium, 20 to 40%, and production traits like lean, tenderness, etc., are the highest there. And so the interesting thing is we know that we can make progress over time to improve fertility in a cow herd. But it's going to be slower than, for example, if we wanted to increase the weaning weights of our cattle, we know we can make that change, genetic change relatively quickly by selecting for larger frame kind of animals there. So again, it's just the way that you look at it, the factors that affect phenotype. If we look at the genotype and environment. So the genotype, the heritability of a specific trait is going to influence the genotype, but also the heterosis is going to influence that, or the additive type effect. And so we'll talk about, or the non-additive type effect, and we'll talk about that a little bit later. The environmental influence on phenotype can be the known factors. So things like, well, if we're feeding those animals enough or we're not, or some of the unknowns could be some of the in utero effects and other environmental concerns that we may not be aware of that could be influences the environment, which ultimately influences the phenotype. A little bit more on genetic side, genetic markers. So a DNA sequence with a known physical location on a chromosome. And so genetic markers are used to track the inheritance of a nearby gene that has not yet been identified, but whose approximate location is known. And so the marker itself may be part of a gene or may have no known function. So, example, a farmer told the city folk, drive until you see the red barn with the right trim on the hill. My house is a quarter mile past the barn, okay? So the marker is like a neighborhood block, and the gene is the house that lies on the block. We just don't know exactly where that house is on the block, but I can give the approximate location. Just like in those directions there, is we're trying to explain where a particular location is. To know exactly, we have to go door to door asking who lives there or what that piece of DNA does. And so sometimes that process of trying to find the exact location is called chromosome walking. And in human genetics, we can see that. They rely less on markers and more identify the actual genes or alleles. Of course, there's thousands of sequences to do, and again, that takes time and money, and over time we will become more accurate to where we can find the specific locations instead of looking for markers or where we think those factors may be there. So selection, the process that determines which animals become parents and how many offspring they produce and how long they remain in the population. There's natural selection all the time. Any of you I know have heard of survival of the fittest. We see that in our natural environments all the time, where animals that survive, they survive for one reason or another, and maybe they acquired a trait that makes them more suitable to an environment. So they're going to be able to reproduce better and produce more offspring and pass those traits on. In man practices, we practice artificial selection. And it's both when we're trying to find a spouse, we're artificially selecting that based off of preference. Same thing in livestock production. We're practicing artificial selection to select those traits, genetics traits, that we feel are the most important, and make that selection, and through time we can improve the population that we have. So the factors that affect the rate of improvement through selection include heritability level. So if it's a lowly heritable trait, it's going to take more time. The selection differential, so the difference between the average performance of the individual selected as parents and the average of the group they were selected from. So, for example, relatively few sires are needed, so they have more effect on the dam. So that's why we typically will spend and purchase a higher quality sire, because it's going to have a bigger impact over time. And that's one of the reasons why we utilize artificial insemination in the purebred side, because we can take that superior animal and maybe breed it to animals that aren't as superior. Economically, we can do that. The generation interval is another factor that plays into it, and so that's the average age of the parents when the progeny are born. So the generation interval on cattle is much longer than it may be in poultry, and especially in broiler production, because it is a very short generation interval in those species, where in cattle we're looking at on a minimum, typical average is going to be about 2 years. Okay? The rate of improvement per year from selection thus is equal to heritability times the selection differential divided by the generation interval. And that's one of the challenges we have in cattle production and selection, is it takes time to make that genetic process, and we're looking at multiple years, where some of our other species, pork and poultry, they can do it rapidly because their generation interval is much shorter. Genetic correlation, something we have to think about when we're talking about genetic selection as we're beginning to mate animals, because often there's a correlation or relationship between production characteristics due to the same gene affecting more than trait. And so these correlations or relationships can be both positive and negative, just depending on the trait. And they may be beneficial or they may be antagonistic. And so, for example, the most common one would be, if I'm selecting for high yearling weight and weaning weight, what would you expect birth weight to do? Well, on average, if we're selecting for higher weaning and yearling, we're selecting for higher growth, we probably would expect birth weight to creep on up as well. And so if we're selecting for very low birth weight, we may be negatively impacting weaning and yearling weight there. Now, within breeds, there are bulls we call curve benders that maybe have a low birth weight but have a high growth rate to enable them to have higher weaning and yearling rates. But on typical, we will see those traits. Again, as we talked about birth weight there. So again, we got to look at those genetic correlations, because they're both positive and beneficial. And if we begin to single trait select something, we have to realize we're going to affect some other production characteristics in those animals. So we have to be careful when we're looking at extremes on there. So some of our selection methods include, one, selection on appearance, which is really subjective, and we still subjectively look at cattle based upon that. And so one of the traits that we really have to subjectively look at is structure in cattle, the skeletal makeup of them, and we'll talk about that because we can't necessarily say, okay, genetically this animal is right, because we know that some animals genetically they may be right, but there could be some structure issues, and they may not be able to breed or just make it survive in the environments we put in them. The other way we select them is on pedigree, strictly basis of the individual's ancestors. And there is some value in that, because we know that maybe some ancestors were better in this trait or another. We select on individual performance, so what the production traits of that animal are. Progeny performance can be more accurate, but it's also the slowest. So we know that if we've got an animal that's got proven progeny out there, there's going to be more predictability in that animal. But it is slow, and we're looking at, for an animal, you're going to look at probably a couple of years on a sire before you're going to see some progeny out of that animal there. And then combining pedigree, individual performance, and progeny performance, putting those three together and taking that data and running it through a statistical analysis, we come up with expected progeny differences, or EPDs, which we will discuss later, and they're very effective with that. And current EPDs are incorporating carcass data, but they're also including DNA data as well into the mix.